and what our mission is. At ViaCyte, we're a leading regenerative medicine company. We leverage our stem cell technology and novel delivery approaches to treat important unmet needs. And our initial focus is type 1 diabetes. And you'll hear a lot more about that in the coming slides. Our long-term vision is to leverage our stem cell technology and our expertise in delivery to treat even other diseases beyond type 1 diabetes. We're really fortunate to have top-tier investors at our side, including Bain and RA Capital and TPG. And importantly, we have 2 world-class partnerships that help us advance our technology approaches. Specifically with CRISPR Therapeutics, we're involved with them on gene editing cells, advancing a gene-edited immune-evasive cell line that we intend to put into patients. And W.L. Gore, which is a leading biomaterial company that helps us with our device innovation and approaches as we combine our device and our cells to treat patients. So that's a little bit about who we are at ViaCyte. As I mentioned, we have deep expertise in 2 particular areas, using stem cells and how to differentiate them, and then once you have the cells, how to deliver them for the patient. Specifically regarding stem cells, as you can see here on the left, we have great IP and deep expertise in the ability to take a pluripotent stem cell that can be defined, differentiated, expanded into a variety of different cell types at the end of the stage. Today, we use our processes to create pancreatic endocrine cells that eventually become alpha cells that produce glucagon, an important glycemic regulator, and beta cells that produce insulin. Now, on the right, getting those cells to differentiate and do those types of mechanical things is one thing, but getting them into the patient is another. This is where we use our devices. We have 2 types of devices today. We have an open device that allows for direct vascularization, engages with the vascular system. That process requires immune suppression today. And then we also have a closed or encapsulated device. This uses surface diffusion to create the membrane approach for hormones and nutrients, etc. These 2 devices are very elegant in their design. They're very slim, about the size of the thinness of a credit card. They're very pliable, and we implant those devices into the patient for therapeutic benefit and effect. We have 2 types of devices, a dose-finding device and a sentinel device. We use a sentinel device to help test the engraftment and get cell histology. So stem cells expertise and delivery expertise as well. As I mentioned, our first major unmet need is type 1 diabetes. Insulin has been a life-saving innovation for type 1 diabetics, and however, the need for finding a functional cure for type 1 diabetes is high. There's a huge unmet need, 1.3 million patients in the United States, 40,000 patients diagnosed every year. And importantly, on the right-hand side here, you can see there is a significant clinical unmet need. There's a high patient burden to count calories, monitor their glucose, and involve that with the activities of daily living. And despite the use of monitors and pumps and injections of insulin, as you can see on the bottom left, there's a very high health economic impact, almost $15 billion a year associated with diabetes and type 1 diabetes, and 8.4% patients actually die of hypoglycemic, so a very high morbidity and mortality. So this sets up the need for our therapy to kind of impact this disease and drive towards a functional cure for type 1 diabetes. What we do with our clinical programs is we manage and monitor various endpoints across various time horizons. Early on, with our sentinel devices, as I mentioned, we can pull those devices out and look at the cells on a cellular basis and stain them, and we can find that we have alpha cells, again, producing alpha cells that produce glucagon, an important glycemic regulator, and insulin, or beta cells, and we get the stains to confirm that we have cell proliferation. From there, we move into biomarkers as the devices and the patients interact with the material, and we track hemoglobin and C-peptide. And then, of course, the rubber meets the road with clinical outcomes. We look at, importantly, time and range, and I'll give you a little bit more information about that in a minute. But importantly, reducing hypoglycemic events, which is the source of a high degree of morbidity, mortality, and cost, and ultimately reducing and potentially eliminating the use of exogenous insulin. Ultimately, what we're trying to do is be in line with the American Diabetes Association recommendations of hemoglobin A1Cs or A1Cs less than 7 and time in range of greater than 70%. This gives you a little graphic sense of why we believe we have a proof of concept for the approach. Starting on the left, and just to orientate you to the graph, what you're seeing here are patient glucose measures across the x-axis of time and the y-axis of glucose. In the gray, you can see that 70 to 140 is considered time in range. So the more you can have the blood glucose in that time of range, the healthier the outcome, both on an acute and chronic basis for patients. This graph on the left represents diabetic patients before therapy, and you can see there's a significant amount of time both above and sometimes below the range. Of late, we've had great innovation from our medical device companies in the market, producing continuous glucose monitors that give insights to patients around what their values are, insulin pumps, et cetera, and they've done a great job in kind of reducing that variability and providing more time in range. However, most of that has been at night, which is a significant innovation for them. But as it requires more activity during the day and glucose counting, carb counting, and tracking their insulin use, the afternoons and the days become more problematic. That has led some patients to pursue and physicians to pursue cadaver islet cell transplant. Here they take cadavers and harvest the pancreatic cells and use a portal vein insertion to patients, which can provide, as you can see, a very functional physiological approach for the patient. They're in time and range much more broadly. However, there's many drawbacks to this. One, you have to take immunosuppression agents throughout your life, and obviously the cell supply is very limited. It's not approved here in the US, but it is done worldwide. We attempt to ameliorate or mimic the cadaver cell transplant model here with time and range, with our endless supply of stem cells and our device engraftment producing physiological response for patients. So our programs, we have two clinical programs. One is, as I mentioned, the open device. That device allows for direct vascularization. Here we've had 33 patients implanted with this device. We have seen in patients, and you'll see in a minute, the kind of data we've seen in patients producing C-peptide, which is a biomarker for exogenous insulin, improved time and range, and as I mentioned, histological evidence of cells. We continue to modify and optimize with the learnings, different surgical techniques, different types of device strategies, and importantly, this is an area where we're going to be using our relationship with CRISPR Therapeutics to use an immune-evasive cell line. So you can imagine an open device with immune-evasive cells that wouldn't require immunosuppression would be a very, very attractive product for type 1 diabetics. Alternatively, we have a encapsulated program, also does not require immune suppression, which is again the goal, to treat the widest amount of patients. Here, this device leverages the membranes from Gore to allow for surface diffusion. And here we're also implanting patients and getting clinical data as we speak. This gives you some visual graphic representation of kind of the results that we've seen. On the left, you can see the evidence of staining that we do for histological evidence of insulin cells or beta cells that produce insulin, and alpha cells that produce glucagon, again, an important glycemic regulator. And then on the right, you can see of the 9 patients who were all de novo negative, did not produce C-peptide enrollment, and the kind of C-peptide, 0.3 as an example here for patient 4, and the kind of C-peptide. This is an evidence that the cells are engrafting, they're surviving, they're thriving, and they're producing insulin for the patient. What kind of meaningful impact can you have? Well, this gives you an indication of that. Take one of our better patients. This shows you pre-implant here on the left, the kind of measurements. As I mentioned, we look at general blood glucose, the amount of hypoglycemic events, and then more importantly, that time and range parameter. 9 months of therapy with our device and our cells, you see that there was a substantial drop in both hemoglobin and also in regards to reducing serious hyper and hypoglycemic events, but really importantly, 74% time in range for this patient. And you can see on the right, that is a meaningful time in range improvement for those patients on a day-to-day basis. So it's a meaningful impact on patients in this kind of patient example of what can be achieved. So we have, as I mentioned, proprietary and exclusive relationships with CRISPR Therapeutics. They're a leader in the gene editing field. Regenerative medicine is one of their growth strategies, and our relationship on our program with them is a top 3 program for CRISPR. Here, we're looking at gene editing our cell lines to make them immune evasive so that they can be implanted into an open device into the patient. On the right-hand side, you can see a few descriptions of our relationship with W.L. Gore. Here, we innovate with them. They are our manufacturing partner, and we leverage their expertise to use novel membranes to mitigate that acute foreign body and giant cell reaction that we often see and we're trying to get past when we talk about engraftment. Broadly speaking, the field has been very exciting. There's been a lot more companies coming into the market. I think that's a signal that this is a meaningful opportunity and a meaningful approach. We're pleased, and I think You can see outlined here how we are the leaders in the field. We've gone from preclinical and into clinical, so from bench to bedside. We have a multiple shots on goal and probably the broadest approach to this market of any competitor in the market. We have 2 clinical phase 2 programs. One, as I mentioned, is an encapsulated program, and one is an open program requiring immune suppression that we would eventually use to put the gene-edited immune evasive cells into. And we're even looking at portal vein and other naked cell approaches. So we're very pleased with the spectrum of programs we have, the partnerships that we have, and the lead that we have in the marketplace. And I think it's a signal about the robust results and expertise we have at ViaCyte. So as I mentioned, here's a spectrum of the kind of catalysts we have coming over the next year or so. Beginning down at the bottom, we're in a phase 2 trial with about 15 patients using our encapsulated product. This does not require immune suppression. There, we're going to have C-peptide and time and range data for this entire group. We need 6 months to do that, so we'll probably be in the 4th quarter of 2021. We had great enthusiasm for the enrollment in this trial, and we're optimistic about the results. The open device, the PEC Direct program, again, that requires immune suppression. There, we had 2 patients enrolled in this trial, then we got hit with COVID. Those 2 patients will be reporting out on data soon, but we are already beginning plans to re-enroll and have quite an enthusiastic response for re-enrollment soon as the pandemic is starting to abate. Here, we're looking at C-peptide and time and range data, and if we have very positive results for either of these programs, I think we can really move forward with the FDA and go for an RMAT designation, which would be a real signal of the efficacy and unmet need that we could produce with our programs. This PEC Direct, let's say, vehicle would also be the place where we would do the gene-edited cells. Here we're using the CRISPR technology, and at the end of this year, we'll be applying for CTA for immune evasion and safety, and then onward in 2022 to an efficacy cohort. I mentioned our ability to leverage our IP and our expertise to not only just go for a big solution and intervention for type 1 diabetes, but also what can we do beyond diabetes. And here on the left, you can see in this graphic how we start with a pluripotent stem cell today, and across those colorful discs, we ended up with endocrine or pancreatic cells that we're applying for diabetes. But really from that 1 o'clock to 4 o'clock zone, here are some of the other organs and disease cascades that we can follow on with. And so we're investigating hepatic diseases, thyroid potentially diseases, and even other diabetic diseases, all leveraging our IP and our technical know-how. So more to come on that, but bottom line on the bottom left here, pluripotent stem cells are unlimited and they can be differentiated into a broad range of different solutions. So we're looking forward to advancing our discovery processes there. Importantly, we have great freedom to operate. We've been in the market a fair amount of time, and we have acquired and developed a significant dominating amount of IP. That IP includes patents that cover how to differentiate the cells, what the cells become, and what the cells actually produce, and even on the left, a combination therein. We have close to 800 patents. The critical patents go out to the late 2030s. So we're really, this is a really powerful item in our ability to kind of freedom to operate and extend into other diseases as well. I think it's important to note that we have expertise and do our own proprietary manufacturing. And so we've done a lot of devices. We've done a lot of bioreactor and seed product cell banks that we've done. We're really the first company that manufactured and implanted pluripotent stem cells at a GMP level. We're currently in roller bottles, moving to bioreactors to be prepared for a scale up to satisfy our clinical needs as we move into broader clinical trials. But it's important that we own, operate, and have leveraged our robust manufacturing process. We do not outsource that today. And then finally, just, all of this gets done, the science and the manufacturing and the clinical trials get done with people. On the right-hand side, you can see that we have great relationships with a wide variety of board members that are both investors, physicians, and experts in the field. And on the left, we have a good blend between people who have been at ViaCyte a long time and folks that are fairly new like myself, who've come from companies like Johnson & Johnson, Abbott, Amgen, et cetera. So the management team and the company is well positioned to execute on our forward-looking plans. The board of directors and our investors are very supportive of where we stand. And as I just would remind you, we have 3 catalysts going with our open device and our closed device and then our gene-edited cells. So I think we're in good position to meet the challenges and opportunities for the future in getting a functional cure for type 1 diabetics. So with that, I'll turn it back to the moderator.
Great. Thanks so much, Michael. I really appreciate the overview and for joining the conference. It's a pleasure to have you.
Thank you very much for the opportunity.
Yeah, looking forward to continued progress and following the data as things progress.
Thank you.
You bet.